Gene Therapy, Transforming the Future of Health One Gene at a Time by Kylie Gabriel. So what is gene therapy? Who engineered it? Where was it developed? And when did the technology begin to thrive? Gene therapy is the transplantation of normal genes into cells in place of missing or defective genes in order to correct genetic disorders. The pilots of this research were Drs. W. French Anderson and Michael Blaze of the National Heart, Lung, and Blood Institute. They worked together to show that cells from patients with a disease called ADA can be corrected in tissue culture. A 4-year-old girl became the first gene therapy patient on September 14th of 1990. She had adenosine deaminase deficiency, more commonly called ADA. It's a genetic disease which leaves her defenseless against infections. White blood cells were taken from her and normal genes for making adenosine deaminase were inserted into them. This method would prove to be unsuccessful for many patients as they would eventually develop leukemia. More research needed to take place. In 2012, the European Medicine Agency approved the first gene therapy for an inherited disease. This drug was called Glybera, and it treated lipase deficiency, which causes fat to build up in the bloodstream. This was the first time gene therapy had successfully been put on the map. Now, let's take a deeper look into what exactly gene therapy is. It's an experimental technique that uses genes to prevent and cure disease. This is done so by replacing a mutated gene that causes disease with a healthy copy of the gene. The improperly functioning gene is inactivated and knocked out, introducing a new gene into the body to help fight disease. Gene therapy can treat ADA, hemophilia, chronic genetic disorders, cancer, neurodegenerative diseases, and other acquired diseases such as HIV, heart disease, diabetes, and hepatitis. So here's an oversimplified version of how gene therapy works as explained by Nick Leshley. Nick Leshley is a biotech engineer and an investor who has served as president and chief executive of Bluebird Inc. in Cambridge, Massachusetts. He's kind of a bigwig. First, make something that can penetrate the 293 T cells, insert 4 plasmids into the cell. The cell then produces RNA, and 3 of the plasmids leave the cell and produce capsid protein, Rev protein, VSVG envelope protein. Therapeutic RNA then encodes the damaged or mutated cell inside the blood stem cell. The virus is packaged and will eventually become the Trojan horse. The virus is actually an HIV-derived virus that has been hacked up and is produced, waiting for the other half, which is the stem cell. Next, you have to go in and extract billions of stem cells from the patient and mix the virus with the stem cells outside the body. The virus then infects the cells and releases the RNA into the healthy blood cell. The healthy RNA enters the cell and reintegrates itself with the DNA. Gene-modified stem cells start producing billions of healthy blood cells. They're put back into the patient, then sieged with a bone marrow, which create what are called daughter cells. Daughter cells turn into macrophages, which cross the blood-brain barrier. And in the case of ALD, these cells act as a sponge, which then stabilizes the disease. This is a picture of a visualization of basically what I just explained. With such an incredible step towards making the world a disease-free place, there probably aren't any issues, right? Wrong. The issues with gene therapy include, but are not limited to, the working gene may be slotted into the wrong spot. People could use this engineering to enhance or alter physical traits in their children, such as eye color, height, and weight. The astronomically high cost could make it the only available to the wealthy. And there are no ethical standards as of current. Deciding who will say which traits are normal and which traits are disabilities, and the fact that it is very controversial still. Speaking of issues, let's talk about the cost. The prices of gene therapy are unfathomable. They could be as high as $500,000 per eye for preventing and curing diseases associated with blindness. Treating hemophilia could be as high as a one-time payment of $4 to $6 million. The options being discussed include a down payment model, which would include annual payments. Gene therapy executives argue that their drug will save the healthcare systems billions of dollars and offer many other unique benefits, which justifies the cost. So, what lies in the future for gene therapy? Well, it's not currently available in the United States, although it is on its way. The first approval will likely be used to treat blindness, and in the future, researchers believe that this type of treatment will cure cancer, HIV, and diabetes. They project that the future of gene therapy will eradicate diseases that have been causing pain, suffering, and deaths for generations. The first FDA-approved gene therapy for cancer may also be on its way. Advexin, made by Introgen of Austin, Texas, may be used in combination with radiation to treat various types of cancer. It's being studied in 2 phase 3 clinical trials. The FDA has designated Advexin as an orphan drug, and if approved, it would win 7 years of market exclusivity. The key takeaways that I hope you learned are as follows. Gene therapy is designed to replace mutated gene with a healthy copy. There are no ethical standards currently in place for the execution of this medical technology. As of current, the price is so astronomical that it would only be available to the wealthy. The US is on its way to approving gene therapy. If approved, this could mean no more cancer and HIV. Gene therapy can save thousands of lives, and there is proof of this across the globe. So despite opinions, it looks like this technology is here to stay. For me, I'm hopeful of a disease-free world that my future family can survive and thrive in. Who knows? Gene therapy just may take us there. I am Kylie Gabriel, and thank you for watching.